Hi everybody, Mrs. Mancuso here, going over some more AP Bio practice questions to make sure you're ready for the test. This is week 7, question 3. Many types of cancer are treated with a combination of therapies. In lung cancer, some tumors respond well to the drug P, followed by radiation treatment. P is a chemical that disrupts mitosis. Instead of spindle fibers originating from the two sides, poles of the cell, P-treated cells develop 3 poles and then divide into 3 cells. Radiation therapy is more effective on tumor cells that have undergone tripolar division than on cells that undergo normal mitosis. Okay, so if I give it a P, they end up doing this. Oh gosh, it's hard to imagine, but they go to 1, 2, 3 poles. So you end up getting 3 cells. So this is tripolar. And tripolar is easier to kill with radiation, or easier to target. And when they target it, they're trying to kill it. So if you have this cancer, you know already that your cells are making tumor cells. If you can get the tumor cells to be tripolar cells, you're going to be better at treating it with radiation if they're tripolar. All right, researchers treated cancer cells in the lab with different concentrations of P for 15 hours. The researchers then determined the average percent of mitotic cells that were tripolar. The results are shown below. Okay, so here the concentration of P. All right, concentration of P is given to me right here. So as I increase concentration, the average percent that are tripolar. So no P, none are tripolar. A concentration of 2, it goes up to 17, and then 48, and then 65, and then 70, and then down. Oh, this last one goes down. So it goes up, 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 up. And then this last treatment actually was a little bit less than the one before it. So it seems like right around here is our best concentration that will cause tripolar cells. All right. And then I don't think they ask a question about it, but I want to point this out. This is my control. You always want to have a control because that is what you're going to use for comparison. And that received no independent variable. So my independent variable was the amount of P. This received 0 concentration of that P chemical. All right, let's move on. The AURKA gene encodes for an enzyme that helps assemble the spindle fibers, which signals the cells to continue through mitosis. When researchers analyzed the level of this protein in different types of cancer cells, they found that cancer cells expressing high levels of this had more tripolar divisions. So increasing the A gene increases tripolar divisions. Okay. They found that cancer cells expressing high levels of ARCA protein had more tripolar divisions. Oh, when treated with Palaxel than did those expressing low levels. Okay, so if you have this gene and you're given the P, then you will have more tripolar divisions, is what that's telling me. All right, I go to the next one. Describe the situations in which a normal human cell would enter the cell cycle and undergo mitotic cell division. So when do cells undergo mitosis? They undergo mitosis when they want to divide. So you want to divide when you want to grow and you want to repair. Those are the 2 situations in which you would find that. All right, the next one. Explain how spindle fibers help ensure the products of mitosis are 2 identical cells with a full set of chromosomes. So what spindle fibers do, I'm going to draw it first, and then we'll talk about how you write it, because the verb here is explain, and doing a diagram alone is not going to be enough to earn points. All right, the spindle fibers come from the centrioles here, and they attach to the sister chromatids. So let me draw this out. First, you can always add drawings to your work. This is my normal DNA. This is what it looks like after it has been replicated. So replication happens during S of interphase, and those line up at the equator and then they are separated. So this used to be here in the middle, and now the spindle fibers attach to the centromere and pull the sister chromatids to opposite poles. And so the question is, explain how spindle fibers help ensure the products of mitosis are two identical cells with a full set of chromosomes. So the spindle fibers attach to the centromere of a replicated chromosome, and then they separate the sister chromatids as the spindle fibers shorten and pull the sister chromatids to opposite poles. All right, this ensures each daughter cell will receive one of each chromosome. So you replicated the chromosome first during S, and then the spindle fibers separated those sister chromatids to each side so that each daughter cell is going to get one of each chromosome. All right, on to the next question here. Using the template in the space provided for your response, construct an appropriately labeled graph that represents the data shown in Table 1. Based on the data, determine the concentrations of P that are most effective. Okay, we always want to have our independent variable on the x-axis. So the thing that we changed was the concentration of P. Again, I would write that all out. I am abbreviating it and calling it P, but you are going to write out the full labels because it said make sure you label the axis. The dependent variable is what you measure. That is always on the y-axis. So here's my dependent variable, always on the y, and I go back to the question, and what they measured was the average percent of mitotic cells that are tripolar. So I put over here, average percent of cells that are tripolar. And I would include the units on both of them. So then what you do, so at 0, it was 0. So you go ahead and you graph each of these. And then the last thing that you have to make sure you do is you put in those error bars. So the error bars, I know I don't have actual readings. You would have to have exact readings and make sure that you have a good scale set up and that you use all the space given to you. So I'm going to just show you one of the error bars. So let's do this one, 17 plus or minus 3. So the way that you do that this would have to be, let's say this is 15 and this is 20. Let's say I went up by 5s each time. To do plus or minus 3, you go up 3 and draw a line and down 3 and draw a line. And so you would get a 1 point for having the correct axes and labels. You would get 1 point for having the correct points plotted and the scale being correct. You get 1 point for having these correct error bars. And then the other part of this question is based on the data, determine the concentration of Plaxel that is most effective. So what is most effective appears to be somewhere here. So it was between 6 and 8. And I think they use NMs. So this is the concentration of P that was most effective. If I go back up to here, there is somewhere in there seem to be the most effective. All right, question C. Based on the data, identify the lowest levels of P that will allow for at least 50% of the cells to be tripolar. Okay, so that's the first thing I have to do. So I go back over here to the data chart that they gave us, and I want to see where 50% is. So I look at concentration 4, and it gives me 48 plus or minus 3.5. So 48 plus 3.5 would give me 51.5. So my answer would be 4. If I went up to 6, that also would give me over 50%, but it said, what is the least concentration of P that you could add that would give you 50% tripolar cells? So you have to include that plus or minus standard error. So it would be a concentration of 4 nms of the polaxyl. Okay, from the start codon through the stop codon, the length of the fully processed AURKA messenger RNA is 1,212 nucleotides. Calculate the number of amino acids in the polypeptide chain coded for by the messenger RNA. Okay, so if my messenger RNA is 1212, I know that every amino acid requires a codon, and a codon is 3 bases long. So the first thing I want to do is divide this by 3. So that would be 404. Now, I know that the last codon to be read is a stop codon, which means it's not actually coding for an amino acid. So I want a -1 for the stop codon. So the number of amino acids that gets coded for would be 403. All right, last part of the question. Predict the effect of a mutation that prevents the expression of the AURKA on a normal cell. Okay, so I want to go back to my thing because I know it explained what that AURKA gene does. So the AURKA gene encodes for an enzyme that helps us assemble the spindle fibers. So I'm not worried about this cancer part. I'm not worried about the tripolar divisions because it says, what would it do on a normal cell? So if this does not work, I will not have spindle fibers. So I want to put in my answer, the effect of the mutation that prevents the expression of this gene would be that it would not be able to produce spindle fibers, and therefore not be able to divide. So if I did not have this gene, I would have no mitosis going on. All right, so that is the end of question 3's explanation.